you'd like the next slide, and I will move it.
Thank you, Alison. Thanks for the invitation. And it's deja vu. Lovely to get back to talk to a lot of people who are interested in the Oceanian coconut production. And for me, I was introduced to the Tree of Life about 1973, I think. And it's a fascinating economy that much of the world doesn't understand. But my introduction was when a colleague was invited who'd been studying lethal yellowing in Jamaica, had been asked to work on kadang disease of coconut in the Philippines. And as he was getting on a bit, he said, I don't want to do it. And I said, well, what can I do if I want to have a play with this new disease? And so I'm retired, but I'm still at university, and I'm going to try and show you a little bit about the viroid disease and also the effect of being involved in coconut diseases, and by introducing the most serious virus disease that I'm aware of in coconuts. Now, the epidemics that I'm going to describe, the kadang epidemic was present in the Philippines. The first indication of disease was about 1917 on the island of Tobacco, and it was on the plantation of two American ladies who noticed that the disease had moved through the plantation slowly, but it was lethal. Coconut tinnigaha, I'll mention that as well, because that resulted in abandoning the coconut production on the island of Guam. And then I'll just mention, I'll show you some of the more recent information on coconut foliar decay disease in Vanuatu. Next, please. So that was the disease of kadang. That was the epidemic which showed up in the central, that's in the Albay province, south of Manila. And it shows a number of features. It shows the crowns, which are obviously dead, and but we have various stages of the disease, but the progress of the disease was slow, but it was inexorable. And so we can have full crowns, reduced crowns, but also the early effects were the cessation of nut production. Next one. This is the current knowledge on the distribution of kadang in the Philippines. These are blow-ups of the mapping of certain areas here, but the disease more or less showed up near Tobacco, and it still seems to be occurring in the central Philippines here. It has not been found in Mindanao. That was a rather exhaustive survey run by the Philippine Coconut Authority in 2012. And that's the extent of our knowledge. We don't know if the disease is spreading as rapidly, but there's still annual losses that are being monitored and checked. Next slide. This is the tinnigaha disease on Guam. Again, loss of inflorescences, reduction of crown, and eventually, whether it's due to a typhoon or just pinching of or penciling of the top of the trunk, the top would just drop off. So that was an economic problem for the local people, but Guam is no longer a major economic or agricultural country anyway. So it's, but it was interesting to get the sequence of that viroid to compare with kadang. Next slide. This is the foliar decay disease in Vanuatu. It first showed up in introduced coconuts. If you look at this aerial view here, we have representation of many introduced varieties of coconut. They were introduced in an attempt to improve production. The local raws and talls produced about a third or a quarter of the expected production. And so the French research station was set up on Espiritu Santo, and dwarf and tall varieties from different parts of the Pacific mainly, but also some from West Africa were introduced. And you can see that vacant rows here indicate that there's been total death. And we see yellowing showing here. We see losses in some varieties, but the main thing was that the local tall variety seemed to be asymptomatic. And so that was the result of introducing susceptible germplasm into a country where the disease was established. And but it also shows the relationship between the proximity of the bush and also the resistance, apparent resistance of the local varieties. Next slide. Next slide. I'll go back to talking about viral diseases now. So the first symptoms of karang-karang in a stand like this is the precocious production of small rounded nuts. And around the same time, the wings of the bracts would show up along the base of the main stem or rachis. There was equatorial scarring on the nuts that formed, but ultimately there was death of the male florets on the inflorescences, and there was and the flowers became, or the inflorescences became quite barren. Severe symptoms were like this, where you had orange or yellow spotting coalescing, but with no necrosis visible. And there was a very severe form identified where the lamina was almost totally absent, and all you could see was the midrib of the leaflets. And so we had diagnosis was based on first of all, the production of excessive production of nuts and the rounding of nuts, the scarification of these nuts, and then the absence of nuts on trees, and then the collapse of the crown like this, where we have reasonably productive palm, a late stage palm, and a dead palm. Now, how do we compare that with Tinangaha on Guam? Very similar setup in the plantations where the tall palms started to show decline. And there were yellow spots developing on the leaflets as well. But the main distinctive feature here, now I can't really delete this little window, can I, Alison? Yeah. I'll move it out of the way. Here we go, that's better. All right, not going. There we go. No, bouncing back. The production of spindle-shaped nuts was the other feature of Tinangaha. And so when we do surveys, we look for these types of features. We look for the production of this winging, the scarification of nuts, and also the aborted nuts here and spindle-shaped nuts. Next slide. One of the outcomes of this was when I first visited the Philippines, they pointed to some of the oil palms, which were showing a similar yellowing in the crown. And these were in the Karang-Karang area. But we soon, as a result of doing some trips around Oceania, and particularly in the Solomon Islands, we noticed that this orange spotting was not uncommon in oil palm. It was called genetic orange spotting at the beginning because it was thought to be inherited. And but now we know that there's quite a reasonable incidence of orange spotting in oil palm, and with the orange, yellow, non-necrotic spots like this, which in transmitted light look like this, but reflected light just look like oil-soaked spots, very similar to what you see with karang-karang symptoms on coconut. A 4-year-old normal oil palm would be bearing like this, and this is the effect. That this is a palm with orange spotting, so they're not producing. They were not producing seed, oil palm seed. Next slide, please. And so this is a oil palm severely affected with a severe strain or severe symptom of orange spotting, no production of bunches. And if you look at the potential reduction in yield, over here we did a bit of an estimate for Malaysia that if the incidence of orange spotting in a plantation was around 1%, we calculated that the annual loss of revenue from crude palm oil was around $25 million. And if the incidence was up to 3 or 5%, or 2 to 5%, you could see we just multiply those factors up. And so we believe that the orange spotting is distributed widely in oil palm, not a high incidence. We don't know much about the spread. We don't know if it does spread, but we know that there is a penalty of infection. Next slide. So the viroids, coconut cutting viroid is an RNA molecule. It's the smallest known infectious agent of any type in biology. It's 246 nucleotides long, and it is a stable molecule. It has no coat protein, so it's not like a virus. There's no protection, but the double-stranded zones through here confer stability to ribonucleases, and so we think that the evidence that the viroid could survive on cutting tools and things like that would explain some of the spread in active plantations. Tinagaha is related, it's about 64% similar to coconut cutting viroid, and it's slightly bigger, about 254 nucleotides in this sequence here. And so this gives us the tool for identification. Next slide. And so a lot of work has been done to inoculate and test and a whole range of palms in the field and also to inoculate them. And we've only found the viroid would infect palms. There are no other monocots or dicots apart from the palms that have been identified as hosts. But if we look at the economic hosts, then we believe then that coconut oil and date palm was shown to be susceptible in inoculation experiments that we did in the Philippines, but we've never found infected date palms in the field. And so economic hosts are coconut oil, and we ask people to check for date palms where that is an economic crop. Next slide is just to mention the work that's currently being done. Most of the work left Adelaide with Ganasan Vadamalai, who did his PhD with me, and he's now in University Putra, Malaysia. And you can see his colleagues who are spread between the University Putra, UPM, and the MPOB, the Malaysian Palm Oil Board, which is involved in using or producing and cloning oil palm for distribution and improvement of oil palm. So you can see now that the methods that are available are real-time PCR and standard PCR, conventional PCR, and also an ISO temperature RT lamp procedure, which doesn't involve PCR procedures equipment. It can be done in a tube using an enzyme which will operate at constant temperature. So the work on diagnosis is now being done in Malaysia. Now let's move on to the virus disease. If you look at the literature, you'll see FDMT as a synonym for coconut foliar decay, was simply because the disease was thought to be induced by the insect, Mindus stepheni. Next slide. And so this insect was shown to be associated with infection by collecting insects in the field, very much in the same type of trial that Richard Davis mentioned, where insects were collected in the field and then put into cages with the susceptible Malayan yellow dwarf. And the only cage in which infected plants showed up was the one in which Mindus stepheni was introduced. And so that work then led to publication of work saying that it was called the disease was foliar decay induced by Mindus stepheni. The vector itself loses infectivity slowly. So these are sequential transfers of the insect onto Malaysian, onto MYDs on a daily basis, and they still retained infectivity soon before, or up until about the time they died. The diagnosis was based on identifying this specific DNA on gels. We could also see a circular single-stranded DNA under the electron microscope, and the virus particles are unusual. They're only 20 nanometers in diameter, and at that time, that might seem, why would I point that out? It's mainly that we didn't know of any viruses which were smaller than 30 nanometers in diameter at the time this virus was first discovered. Next slide.
5 minutes for the end of the presentation, John.
Pardon me?
5 minutes for the
5 minutes to
yep.
That's good.
Yep.
Thank you. So this is the vector. So the larval stage and adults all develop on Hibiscus tiliaceus. The females, so the male females and males are easily identified, but it's unusual in that this particular species is localized only in Vanuatu. You'll find Mindus in Solomons and in Santa Cruz Islands, but they're different species. And so there's been no cross hybridization of the vector. So that's what we know about the distribution of the vector. It seems to be just in the Vanuatu archipelago. Next slide. Coconut palm is the only known host of coconut foliar decay virus. Mindus stepheni is the only known vector, and the larvae are found on the roots only of Burao or Hibiscus tiliaceus. This is the common and right through Oceania. The adults, though, are found on the leaves of coconut palm, and feeding produces the disease in coconut. So with this information, control is directed at removal of the Burao and resistance and resistance breeding programs. Next slide, please. So hibiscus growing in a plantation, very common plant, and the larvae are found on the roots. The larvae develop into adults, and then we find that the vector is in the plantation, and we can see the effects of the vector where the trees have had their crown has just died. The disease takes about 8 or 9 months to show the first yellowing after inoculation, and in the most susceptible varieties, Malayan dwarfs, the crown usually is dead within 2 years after the first symptoms appear. So the control is directed at identification of the virus these days, diagnosis by PCR, and also we can simply make a hybrid, a probe labeled with a radioactive or some other non-radioactive nucleotide, and use that for diagnosis. Next slide. And so when we take insects, males and females here, we've just squashed the guts onto a nylon membrane, and we can probe them with a labeled probe, in this case radioactive, and both the females and the males have detectable virus in them of the adults. Next slide. And we can also do northern blots where we can identify that it's just a single nucleic acid, single DNA present in positive insects. So not every insect is positive, but there are strong signals for the presence of the DNA indicating that they're potentially positive vectors. Next slide, please. So CFDV is unique. The virus has only been found in Vanuatu, and coconut is the only known host. And the vector, it's only lethal to introduce varieties. So the current control is to hybridize introduced dwarfs, or hybridize the local dwarf with the local tall. They seem to be the most productive and resistant variety. The vector has only been found in Vanuatu, and the virus itself contains 3 genomic circular single-stranded DNAs, which are unrelated to any other known virus. But most recently, we've discovered that in the virus are some so-called alpha satellites. So this means that in the virus particles, we can have 3 DNAs, one which will code for the polymerase, one will code for the protein, one will code for movement protein within the host. But also we found a lot of other DNAs which are similar in sequence, but don't have any function, except that we wonder if these have some function in modifying the expression of symptoms. But this is a new finding related to virology, probably not of great interest to other people who are interested just in other organisms. But we have now set up a new family for the virus. It's in the genus Cofodovirus. It is the only virus in this genus, and it's also the only virus in the family Viroid. So it's given us food for thought in the world of virology as well. So let me summarize. So coconut palm has been affected by a number of historical epidemics, and that's why these agents have been found. Karang caused losses of around 40 million palms overall in the Philippines. It's the smallest known pathogen of any organism. It's currently controlled by cultural methods based on molecular diagnostic methods. We still don't know how we've never found a real vector. We know that you can transmit the virus through pollination, but the rates are quite low. And oil palm is a newly recognized economic host of Karang Viroid, and I think this is the reason why it's important for Malaysia to take over the role of developing studies on epidemiology and control of Karang Viroid. Tinagaha has basically caused the loss of the industry in Guam, but you can still find trees, and the material that was sequenced was collected by a colleague, Bob Simons, in Guam back in the early 80s. And so that's been a great benefit to understand the relationship between these 2 viroids. Foliar decay has prevented genetic improvement of coconut in Vanuatu, and so it's it also is a unique virus with a unique vector and also with associated alpha satellites. Last slide. This just summarizes the positions where the localizations of Karang in Philippines, Tinagaha on Guam, foliar decay in Vanuatu, and this red line right round indicates the zone where we've been surveying monocots of various kinds using northern blot techniques, northern and southern blot techniques, and we found within that zone the viroid-like sequences. That is, we can find short, small pieces of nucleic acid which will come up positive with our probes, but we have not been able to follow that up any further. That's for the future. So thank you very much for letting me talk to you about my favorite tree.
Thank you very much, John. I think it's a favourite tree of many people today. It's a wonderful presentation with lots of information, and really nice to see those examples. Well, not nice to see those examples, but nice to see examples to illustrate your points. And they're obviously quite, very serious, viruses and viroids, when they get a hold in a palm plantation or in a coconut plantation as well. So I've got a couple of questions, and there's a question here as well. I'm going to read it out. That Padang disease was in existence in the Philippines for more than 6 decades, and so far cut and burn is the only way to eradicate the disease. Are there any other options to mitigate the disease spread despite its slow rate? It's still surveyed in coconut growing areas. With the impact of climate change, is there any possibility that there may be some increase in its spread, considering that massive planting and replanting is underway in the Philippines?
Yes, replanting is the answer for this. The early studies on the epidemiology showed that the infection disease was moving very slowly, and plantations often didn't show disease until they're around 10 years old. And so the policy these days is to take productive hybrids, and if there's a diseased tree, any diseased trees are cut down and they're replaced with a precocious F1 hybrid usually. And there's an arrangement, as far as I understand, between the Philippine Coconut Authority and I don't know the arrangements. You would need to talk to them about the current arrangement, but the policy is to replace any trees with symptoms with productive hybrids. And the hybrids themselves are usually now given a life of only 20 years or thereabouts, compared with the olden days when the tall coconuts you would have seen 50 years ago, they would have been up to 80 years old. And so in some ways, cutting was showing up as a result of having aged tall coconut palms, still productive, but the surveys showing that the disease was slow moving and not showing up until new plantations were put in until they're at least 10 years old, leads to use of cultural control measures. The identification, well, the molecular identification methods now are available, and I have not had contact with anybody who's doing the setting up with a lab in Manila, but I understand the research station in Albay province, the so-called Ginabatan experiment station, or the Albay Research Centre in Ginabatan is still the main research centre where information is available and where any work is being continued, and that's within the cutting zone.
So here's a question here, John. Thank you, John, for the informative presentation. My question is, are there any effective treatments or management strategies for palms already infected with a viroid or virus?
No, I understand that they're simply cut down.
Yep.
And we do know that the husk of dry nuts, the viroid is stable on dry dead tissue for some time, and I would say that destruction is probably by burning.
Yep. So there's no way of treating it and recovering. It is just, you need to get rid of it.
I think that's what I would think. At the time the project that the FAO and ACO project was set up, there was an attempt to utilize coconut wood from trees that were cut down, but I don't know if coconut logs are able to be transported outside the cutting zone.
Yeah.
And
so the last question, I think, John, is, I mean, you've given 3 examples here, and it's quite surprising because they're quite unique examples and quite specific locations. What do you see happening in the broader region? I mean, are there other examples or serious types of viroids or viruses? Have we just not found them yet? I mean, hopefully that's not the case, but you know what? They seem very unique, some of those examples. So what, have you seen anything that's alarming? Or I guess you gave the example of Malaysia with palm plantations, keeping an eye out there. Is there any other examples in the region you've found, or you think could be areas that need close attention?
Well, Sri Lanka would be the main country which is concerned with a number of diseases which have been given names like rapid decline and lethal decline, but the agents have not been found there. We do know that Karang Viroid has been found in non-bearing dwarf trees in Sri Lanka, and we did set up a lab there for a while. The other diseases are of concern in Sri Lanka, but I think that the basic work, that is an advanced lab, needs to be set up with labs and with control with trees, and putting a big effort into tracing the nucleic acid components. And I think with microbiome analyses these days, with high throughput sequencing, next generation sequencing, I think this may be the next approach. Is going to be productive, in that disease trees can now have a whole genome sequenced and then analyzed using next generation sequencing methodologies, so that the microbiome of these trees can now be established. That is, to subtract the host nucleic acid sequence away from others, which could then be checked under by gene bank and in blast comparisons with known pathogens. It's just that coconut palm has been a real Cinderella crop in terms of, it's been a number of diseases and pests that have been described from observations externally, but finding a virus and a viroid were quite unexpected symptom or unexpected events. But we wouldn't have started work on these unless there'd been epidemics of those diseases. When we do surveys of countries, we need to go to a place where there's a plantation and there's a known described depression or unusual event where there can be individual trees are not performing to the expectation. And then the I think once we have these basic sequences available, then we can at least use those in a gene bank situation where we can say, okay, we've tested for these. But I think next generation sequencing, now that it's becoming a much more powerful methodology, that any new work, I think, should be using that.
Excellent. Well, that's probably a good place to leave it on, because our next speaker is just going to follow up from session 1 that we had a few weeks ago, and he's an expert in genomics. So it's probably a nice segue to him, John. But thank you so much for that presentation. Really very nice, and really nice to see the examples. And I think also there'll be potentially opening up of a lot of new research with these new tools in the next coming years, but it would be nice if they stay uniquely in their unique locations, I think, rather than spreading around. So thank you very much, and we hope to have you back at some point for an update, and good luck with your work.